So I'd like to make this informal. I'm going to run through a number of things that I think are going to be important, not only from regulatory aspects of developing drugs, but also about timelines and costs and so forth. And I'll heavily qualify the costing part of it because I'm going to give an example. It's probably the most inexpensive example that you can do for a small molecule. It gets a lot more complicated for large molecule, but I will comment on that. I also like to be informal enough that if you see something that I either haven't explained or I violated my own rule, which is don't use an acronym without explaining it, please raise your hand and just stop me, and we'll take care of it right away. All right, so without further ado, so this is a very simple table that talks about the various stages from drug discovery all the way down to drug production, and the various phases that have to go with that. So in the early phases of drug discovery, you're doing many things both in vitro and in vivo, and you're also doing them as preclinical animal studies, and they are not necessarily going to be GLP compliant. So GLP is good laboratory practices. So anything that the FDA requires you to supply to them usually have to be GLP, but there are some exceptions. And in some cases where you can't validate an assay or you can't, it's not an interpretation changing part of your study, you can just declare it non-GLP. The no-no is to have them find out something that's non-GLP and ask you why you didn't declare it. So if you're upfront with them, it's usually smooth sailing. So then we'll talk about, oops, I hit the wrong button there, didn't I? We'll talk about the FDA regulations, the difference between GCP and GLP regulations. So GCP is good clinical practices. You also hear about GMP, which is good manufacturing. And another acronym that I will probably use that Peter Kuritko, if you haven't met him, and I have done on various drug development symposia, is CMO, which in some companies means Chief Medical Officer, but in the chemical manufacturing world, it means Chemical Manufacturing Organization. So it's like a CRO, but it's for manufacturing. And that's to do with regulations called CMC, Chemical Manufacturing and Controls. So there's a little bit of explanation going right into it. So you see GMP regulations here, so that's for formulation and manufacture. Often where companies we deal with run into problems are that they can make their drug, but they can't formulate it, or they can't do a fill and finish product that has to actually be given so it mimics what's going to be done in the clinic. And as you'll see later in the talk, I focus a lot on how we set dose levels based on what you will do in the clinic, because that's exactly what the FDA is going to ask you to do. I speak a lot about the FDA, but in fact, the guidelines that most people follow are ICH, which is the International Conference on Harmonization, and FDA is a participant in that. When I first started my career in toxicology 32 years ago, there were different regulations for different theaters. So Japan had their own regulations, Europe had their own regulations, and this was a movement started in the late 90s to consolidate all of those and find the common denominators and list things that were not common but needed to be thought of. And so that was organized under ICH. So you'll hear me say ICH on occasion as well. So here's ICH, and it just shows the different theaters. So North America, Europe, and Japan, regulatory aspects and industry, and they were all combined under one set of rules. The other thing to note is in the regulations, the FDA was started in 1938, and it was started to regulate foods. It really didn't even have much to do with drugs back in that. But that was called the Food, Drug, and Cosmetic Act. And so many of the things that you see on TV today, where you see that little disclaimer at the bottom saying this has not been reviewed or approved by the FDA, that was pretty rampant back in 1938. In 1984, we passed the Hatch-Waxman Act, and that was for generics. But what they didn't cover was large molecule. It was all small molecule focused, and we'll talk about that in a little bit. And then in 1992, we had the PDUFA Act, which was the Prescription Drug User Fee Act. And what that was designed to do is speed up the FDA review. So you pay some exorbitant amount of money, $250,000, and that was supposed to speed it up. You know, presumably so they could pay people overtime, et cetera, but it really didn't have the effect that they intended. So that's a little bit of the regulatory background. And then, of course, back in the day, FDA was divided up into CDER, which was small molecule, CBER for biologics, and then other things were food, cosmetics, and supplements. So that's a little bit of the history behind that. This is the PDUFA Act that you can go online in the Food and Drug website and take a look at. And I'll leave that for you guys to find your way through if you are interested in it. Then we get into preclinical trials. Okay, so these are the studies that laboratories like mine will conduct for you in order to support what you want to do in the clinic. So you're aiming at this stage of the game for what you're going to do to support phase 1 clinical trials. So with the exception of cancer drugs or some of the orphan drug indications, you're typically going to be testing in what we refer to generically as normal healthy volunteers. Okay, so you're looking for effects on people that don't need to take the drug. Is it safe for them? That's where you start in the clinic. If you're in cancer, you go right into cancer patients in phase 1. And where you have a control or placebo group in phase 1 for all other drugs, in cancer you have a comparison with the standard of care. And what you hope for in cancer drug development is that by the time you get out to phase 2 and 3, which are the later, bigger stages, and you'll see that in a minute, that the FDA tells you to stop treating with the standard of care and put those patients on your drug. You've hit a home run at that point. Okay, so again, a little bit of oversimplification, but this is the animal testing that we will start with in drug efficacy studies, and then the preclinical testing, which also includes safety testing, safety pharmacology. So this goes through short-term and long-term, and so you're building towards an IND application, and what this does is summarizes your animal study results, your manufacturing results to that point. It's a sort of a progress report, and your clinical protocols. What you intend to do to put your drug into clinical trials. And this is your investigational new drug application. The FDA gets 30 days to review what you submit when you submit your IND. And you don't have to wait for them to give you the okie dokie. If they don't get back to you within 30 days, it means they don't have any questions. Yes, sir.
So the in vitro studies don't go into the IND application at all?
It depends on whether or not they're pivotal in terms of proving a proof of concept, or this is showing that my drug will work. But they don't go in any form that's a standalone. Remember, in vitro, in my mind as a scientist, signifies an idealized condition. It's not whole body. And so they will put a lot more emphasis on the whole body results. So it's fine to supplement, but they're not standalone results, if that makes sense. Yes, sir?
What level of animal work is necessary? Does it have to be in primates? Or
we'll get to that. It depends on your pharmacology. Okay, so if your drug's not active in dogs and it's active in primates or mini pig, those are the 3 main non-rodent species. That's what drives what you'll do. And I've got a couple of examples I can give you where we had to make a decision like that or help the client make a decision like that. Good question, though. All right, so this is not intended for you to study in detail, but I have an example I'll pass around. And this is a timeline that you can just pass that around so you can see that in more detail. This is a timeline that we give to our clients that shows that for a small molecule, we can produce the reports in final form that you need to submit for your IND to the FDA in less than 6 months. And that's a pretty rapid timeline. We've gotten a lot of compliments about this. You'll see on the back side as you flip that over that we've made some suggestions as a small company what you can initiate in terms of other studies post-IND to support phase 2 and so on. And those are really good things to think about if you're going to partner with somebody else. The longer you hold on to an asset, and you'll hear venture people talk about this, the longer you hold on to an asset, the more valuable it becomes. But most of the people that you want to either fund your studies or partner with you are a little bit one-sided. They want you to vet all the risk. So the longer you can hang on to it and the more money you have to actually conduct these studies, the better it is for you over time. Now this example gives rat as the non-rodent and dog as the rodent. And we show 4-week studies and a 4-week recovery in each case, and we're still hitting the 6-month target. So back to the question we had about non-rodent species, the dog is important here but that may be because of the pharmacology of the drug. It's not active in any other species. I tell people two things that I've found to be true over my career that you need to pay attention to, and that is that the FDA does not care how much it costs you or how long it takes you to develop the drug. That's not their charge. Their charge is, because they're organized under public health, it's safety. It has to be both safe and effective, not just effective. You know, most people that are focused on the funding, they say, well, if you don't have efficacy, then you're not going to get funded. Well, that's true. But what I'm telling you is if you don't have safety and you haven't shown it, you're not going to get past the FDA. So this is a slide that I've actually used in a CMC symposium, so I concentrate on helping people determine what they need to do proactively for their drug manufacture. And you can see that right here, there's a very small window about when the drug arrives. That's usually about 3 to 5 days ahead of the very first study that we would conduct for you. But if that doesn't happen, you know, the whole schedule slides, because these things are designed to be, they're in parallel, but there's certain things that have to happen first. So you have to do a dose ranging study to set the doses for the definitive studies. And what you'll do in the clinic will either be dictated by a 2-week or a 4-week study in the IND. Okay, so that depends on what you're trying to show in the clinic. And remember, in the IND, the last part of those 3 bullets under the IND was, what's your clinical plan? What do you plan to do in terms of dosing patients? It doesn't have to be definitive. It doesn't have to be, this is exactly what I'm going to do, but you do need to show them a plan showing that you've thought about it and this is what we're going to do. Okay, so in talking with Craig Phillips, who I see is in the last row, he thought this would be very, and if you don't know him, meet this guy. He's a mover and a shaker. He thought it'd be very good if I gave you an example of what would be both the timing, which I've started here, and the cost. So this is what a document coming from our laboratory looks like. And I've just entitled it, or we've just entitled it, Standard IND Enabling Package Rat and Dog. Okay, so on the 6th page of this document, you can't read this, but I'll blow this up in a second. We line out all the studies that will be required for your IND, and we give you pricing for it. And then if you blow this up, can all of you see this? Is this, no? So this is an example, and if anybody wants a copy of this, Craig asked me if I could provide this. I certainly can, and I can do that electronically. But this is just an example of what your studies would cost, and hopefully you can see this. It's about $533K for just the 4 pivotal studies, or dose-ranging studies, and the 2 definitive, or what we call pivotal IND support studies. These are 28-day studies again, and then the whole program runs to about $887,000K. Now your price is going to vary depending on several things. What's your route of exposure? Are you going to have to do something exotic like subcutaneous, intravenous infusion? How long is that going to be? All those things dictate how difficult or complex a study may become. So that's what goes on there. And then with each of these line items, we give you a full study outline so you can see the detail of what we're providing the cost for. And then at the end of this, I don't have a slide, but at the end of this, you'll see options that could be included. So we give you a base price, and then we tell you what the options will be depending on whether or not you want to include them. I'll send this document around. I've got one for each side in this case now. This is a very popular document that were written by 2 of my colleagues that joined us from Eli Lilly. I affectionately refer to them as Lilliputians. But they wrote this document, this booklet, and I can provide this electronically as well to anybody who wants this. Just give me your business card at the end if you want a copy of it. But this is actually a very good compendium of what to do and, more importantly, what not to do. So it's a points to consider type of document that's organized by therapeutic indication. The only thing it doesn't cover, and I don't know if there are any medical device people in the audience here today, but it doesn't deal with medical device. That's a whole different set of regulations, and I'm not going to cover that today either. So that's kind of where your pricing is for small molecule. Look at this from a Tufts University study done in 2006. This I actually captured from a website of a very good friend of mine who's a consultant in the Bay Area who deals a lot with large molecule because he comes from Genentech. That's his background. And the tagline he has on his website is, would you like to do better than this? So faster and less costly. But this actually is a price that encompasses all of the clinical testing that you're going to have to do to get your drug to the market. So it's pretty formidable. So now this is an idealized timeline for large molecule INDs. And what you can't read here, but I'll just tell you, is this is all analytical formulation and bioanalytical development, because you need to be ready to measure samples as they come out of the toxicology studies. Many people start their small molecule studies at what we call at risk, because they've got analytical information, or at least stability information that says I can freeze plasma samples and analyze them when I can get to them. So they start the studies and they validate the study on the way there. Now, one of my favorite words to ask people in this industry is, what do you mean by validation? Because everybody's got a different variation on that theme. But in the eyes of the FDA, what this means is you can show robustness, reproducibility, and the ability to cover a dynamic range of where the concentration might be in the samples that you harvest, okay? And at this stage of the game, you're only talking about study animals, not clinical samples, because you haven't gone into the clinic yet. And that dynamic range may change. But the validation is actually important, because if you change laboratories, if you change contract labs, in other words, I started one study here, but I'm going to put another one over here, you have to revalidate that method in the other lab. And we've had to validate methods that we've had at our State College, Pennsylvania facility when we transfer them back to Kalamazoo, Michigan. And that's just the rules. The FDA is very, very strong on that. So there's the test article arriving again, and you can see it's a very small window in the grand scheme of things. So here, and this is the other thing I'll leave you with, and there are a lot of, I'm not permitted to give this out because there are so many caveats at the bottom of the page. So all of these footnotes are, okay, if this happens, then you got to do this. And it just gets very complicated, and people got a little nervous that I was giving this out as though this were the Bible, and I just don't do it. But I show it to people because the emphasis is really that the testing is actually almost a half a year later, and you're doing this. These are 14-day, these are mouse and monkey, I believe, NHP, and you incorporate a lot of the endpoints that you would do as standalone in a small molecule into your study design for large molecule. The expectation is that most biomolecules are not going to cause safety pharmacology endpoints to be tweaked, if you will. So what you study as a core battery for small molecule in cardiovascular safety, in CNS, and in pulmonary safety are all combined as measurements you can do in the large animal, because they're expected to be no effect by the compound, okay? There will be exceptions, and I have had people do these studies anyway, just so they have them in their back pocket. But if you have anything that borders on GI disturbance or renal clearance, you know, they often will elect to do those just so they can say, we have looked at this independently and our compound doesn't affect that. But the big thing here is timing. How much longer the time course is. This is an entire year depicted here versus a half year on the other one. And all the caveats that you have to negotiate ahead of time in your pre-IND meeting. So it gets very complicated. Any questions so far? Okay, so this is the conundrum of any contract lab. Quality, cheap, on time, pick two. I saw this on a sign outside of a military laboratory that I was interviewing for at Wright-Patterson Air Force Base before I took this job. And it really says that this is the most difficult part of what I sell, what I do. So most people would like to, oops, go back here. Most people would like to focus on quality and on time, but they also want cheap. They also want inexpensive. And oftentimes what I do is I walk them through what the expectations are and where they can't cut corners. Now, I try to cut corners where I can, but I also try to be upfront with people so that they don't think they're getting a Cadillac for the price of a Volkswagen, I guess is the way I put it. And it's a tough aspect, a tough thing to juggle. That, by the way, is a picture of the University of Michigan students reacting to being beaten by Appalachian State in the first year of their coach's existence. So that was Rich Rodriguez, who I think they ran him out of town on a donkey. They didn't like him. He's actually now the coach at Arizona, which is really interesting. So I'm going to go over this slide very quickly. This is phase 0 clinical trials. And this is often done for exploratory INDs, which are fairly rare, okay? But this is where orphan indications often fall. If you have a life-threatening disease, we have a Bay Area sponsor that's working on a mitochondrial disease that kills young children by the age of 5, and they've had fantastic results. And they have literally an upside-down IND. They're doing all their long-term stuff first, and now they're going back and backfilling. So this is a designation for exploratory first-in-human trials. It's microdosing. You'll often see people do this when they have an imaging agent, where they're going to give a very, very minute dose, and they can go this route. Again, designed to speed up development. Does a drug behave in humans as expected from preclinical studies? So you're going directly into humans with this, with very few animal studies having to do. It's a very small number of human subjects. You're usually administering, a small, very sub-therapeutic dose one time. And so it's kind of the exception. And it does not study safety or efficacy, only human exposure parameters. So it's a very qualified, designed study. So we get back here to clinical trials and we start talking about the longer ones. Okay, so phase 1, the emphasis is on a small number of volunteers. Oh, I'm sorry, thank you.
Phase 0 studies, when you say human exposure parameters,
Right.
what sort of things has that turned out to be useful for? PK mostly?
It's mostly PK, exactly. And if you think about imaging agents where they're giving, very, very minute doses with, large doses of radioactivity, they're just looking at distribution. Where does it go? How fast does it clear? And again, these are negotiable. There are also 2 other, that's exploratory. There's a second EIND, as we call them. It's called emergency IND. And this company that I talked about in the Bay Area actually did an emergency IND with one patient, 1-to-1 with a physician. And that gets really weird because the physician has to pay for the study. They can be reimbursed by the company, but the FDA only wants to talk to the physician. It's a very strange and rare exception.
In the last couple of years, there have been 75 drugs, last 3 years, 75 drugs approved. How many of those drugs went through a phase 0, or is it not public information?
I don't think it's public, but I would say probably a handful of them. Yeah. Somewhere between 1 and 5, maybe. The vast majority of them are going to go through the whole gamut. So again, as far as numbers, thank you for catching that. I appreciate that. As far as numbers, you got a small number in phase 1. You expand to 100 to 300 in phase 2. And you'll usually hear people talk about phase 2A, phase 2B. That'll be for different indications, different endpoints. And then you start getting into thousands of patients. And sometimes in things that affect congestive heart failure or large portions of the population, you might be doing clinical trials that border on tens of thousands of patients. And that might be a requirement, and that's where you start to get into that 1.2 billion that I showed earlier. And again, those are estimates that were collected over time and only as good as the information they had in 2006. So I always caveat that. There is such a thing as accelerated approval, okay, and post-market surveillance. So rare adverse effects are probably the single event that kills a blockbuster drug. Big companies hate this because it's an idiosyncratic reaction of somebody with liver toxicity or something that's life-threatening. And in some cases, 3 or 4 people die and the drug is pulled off the market. I think we saw that with, not Ambien, that was a sleep drug, but the one that, Celebrex. Now that got pulled off the market and then it got petitioned and now it's back on the market. But that was a case where I think only 3 or 4 people died in the grand scheme of thousands of people that took it, okay? So idiosyncratic reactions are really the bane of blockbuster drug development. And then this accelerated approval is based on unmet need, based on patient populations that won't be served if you just leave it to the big companies to get around to it. So there's exceptions. And I can tell you, having worked for 2 of the biggest pharmaceutical companies, there's a lot of drugs sitting on the shelf, ladies and gentlemen, that just never got developed for strange reasons. An Ames mutagenicity finding. Well, we just put it aside and we go on to the next one, It was probably a damn good drug. In fact, I have some friends that were in the pharmacology division started a company for ALS, Lou Gehrig's syndrome, and they brought their drug from the first toxicology dose they did with us to phase 2A in 15 months. That's cooking. But they decided that they were going to get out of the big company and do things better. And the reason they went that fast is because they made decisions. When stuff comes up, and there will be adverse reactions, there will be things. That was a case, back to the non-rodent question, couldn't do this study in dogs. It was a D2, D3 agonist, and it caused severe emesis, so your exposure was all over the place. And the other thing you'll hear me say, if I don't say it again, is that when I give you a toxic dose, because that's what the FDA wants you to look for, I express that in milligrams per meter squared, because that's the conversion factor to humans. They want to know what's going to convert from animals to the clinic. Okay? They don't care about milligram per kilogram, because that doesn't tell them anything about exposure. So in that case, we couldn't give it to dogs, and we found that if we gave it to Gottingen mini pigs, who are about the size of a large dog, they're about 10 kilos, 10 to 12 kilos when you start, you could go up almost 3 times higher in dose before you see emesis. You really had to push the dose. Yes, sir?
So for drugs that are repurposed,
Right.
that would be tried in different disease,
Right.
where do you have to start this process?
Well, you have a lot of data that you can call back to. And so some companies will take an older drug and do that, but they'll do what are called bridging studies. And you see the generics do this too. One of the things that the big companies hate is the little generic companies will grab a drug and make a generic form of it, and all they have to do is prove that their drug is equivalent to the innovator drug. Okay? So what big companies have done, to your point, is they've started to repurpose. They've started, they had a great term in Bristol, it was called life cycle management. And they've formed their own generic companies. So they're trying to beat the generic companies to the punch. And I've got an example in large molecule that will floor you later, because it was really, nobody saw it coming. Did that answer your question? It's a lot shorter path, but what I always tell people when they ask that question is go back to the summary basis of approval, because every drug that gets approved has to have a summary basis of approval document from the FDA that tells you what they base their approval on. But be careful about the age of that SBIR, because if it's over 10 years old, maybe even 5 years old, it's probably got information that's changed. You know, they're thinking differently about this, okay? PPAR agonists, I think, are probably a good category to look at, because that's changed. When I was coming up through the chemical industry, PPARs were considered a classification of rats only. Didn't have to worry about them in humans. And guess where we are today? You now have to start, if you're in a PPAR agonist development, you have to start your 2-year carcinogenicity studies before you file your NDA. Okay, in some cases they'll let you file it afterwards because it's considered post-market surveillance, and it depends on whether it's life-saving or not. Okay, so by the numbers, this is just kind of an interesting slide. Again, it's a little dated, but discovery to preclinical can often take 2 to 12 years. This is probably based on big company data, so it'll vary depending on your category and so forth. But you're spending around a little over $500 million in screening about 5,000 candidates. Your clinical trials will take 6 to 9 years, and you're doing about 5 candidates that will enter trials for one successful candidate, in other words, that will go on beyond phase 1. There's another little over $500 million. So the total capitalized cost is $1.2 billion. So that's where the number came from, okay, that they put in the tough studies.
So it's including all the things that have been tried and failed in that?
Correct.
All right.
Yes. Okay, so again, phase 1, what are we looking at? Healthy volunteers receiving escalating doses until side effects are seen. So again, you're establishing what's my threshold of toxicity now in humans. You've done that to get here in animals. And you set your dose levels in the clinic based on what you've done in those preclinical studies. This establishes the maximum tolerated dose, okay? So the high dose there is the one that makes everybody unhappy, including the investors. It always cracks me up to see investors panic when you demonstrate toxicity, but that's what you're doing these studies for. And it's not, they don't want to tell their board of directors, they don't want to, and it's like, no, the FDA wants to know this. That's what they make you do these studies for. Okay, often patients take part in phase 1, and that's oncology, and I think I mentioned orphan drugs and some other specialized categories. But typically they're not patients at this stage of the game. And they continue all the way throughout development. You tend to think phase 1, phase 2, phase 3, and then you even talk about phase 4 for the post-marketing. But you're done with phase 1 once you conduct a phase 1 trial. No, you're doing that all throughout development because you're changing your formulation, you're changing the dose form, you're maybe changing the route of administration. All those things have to be reclassified in phase 1 studies so that they can show, whatever company is developing, can show the FDA that you haven't done anything to harm people that don't need to take the drug. Yes, sir.
What's the value of the second, establishing maximum tolerated dose? You know it's going to have an effect, so that doesn't really mean anything until you know the amount you need to actually get efficacy, right?
In the clinic, what you're trying to show is that that dose is going to be far higher than what your efficacious dose will be to actual patients. So you're now testing in people that aren't affected with the disease classification, and you're trying to show that, okay, when I hit this threshold, this is probably where I'm going to give the drug in the clinic. So I've got 100-fold higher dose multiple to establish toxicity in humans.
But in phase 1, you haven't done efficacy yet, right? So how do you know?
But you have, based on your pharmacology studies, you have an idea of what you want to give. And you've presented that in the phase, or the clinical plan as part of the IND. So this is kind of a back check to that. It's a good question. They expect you to talk with them regularly all throughout this development. Because, they tell you, and that's what PDUFA was designed to do, that they want to partner with you. Now, people will tell you horror stories about whoever they get, because some of them are real so-and-sos. But in my experience, and this is why I tell people I give free advice, I don't charge you for consulting, but my experience is CNS and infectious disease. If you're not in those two spaces, I'm not probably going to help you very much. But I know a lot of consultants that can, and that's what you pay them for. You pay consultants to have subject matter expertise and to know the players in the division you're going to at FDA, so there's no surprises. And the really good ones, when they present the IND, will say at the end of it, we would like to hear what concerns or issues you may have with the plan we presented. What I always tell people is don't go in there saying, what do you think we should do? They'll send you home. And typically nowadays, you don't even get to go to have a pre-IND face-to-face. It's all done by teleconference for the most part. And the best INDs are when they send back, we don't have any questions. Proceed. And that's directly related to how well you've done your homework. And typically whether the consultant you've paid money for has earned his or her pay for that. Again, it's cheap insurance because this is tough. This is a hard field to negotiate. I say that you pay a consultant to help you negotiate the minefield at FDA. It's really true. So then we get to phase 2. These are a larger number of patients like we talked about on the previous slide, but these are all patients now. Larger number of participants, pardon me. The drug efficacy is tested against a standard of care or a sugar pill. Okay, so standard of care is typically cancer because it's not considered efficacious to test a placebo on a cancer trial. And then you get to phase 3, the bigger studies. These continue on a much larger scale. They're randomized and blinded so that the conclusions that are drawn have nothing to do with knowing whether your patient got a drug or a placebo. And then the drug treatment sometimes continues after the former end of trial. And that's usually in compassionate use categories. So you'll see that on occasion. Okay. So this is just another slide through 2006. My data is not current in this decade, but these were available data at the time I first started putting this talk together. And you can see that of the new molecular entities, very few up to 2006 were large molecule. And they're much harder to get through, and they're also harder to make bioequivalents, the Biogenerics as they refer to them, or biosimilars. I've even heard a term called bio-betters. So.
Why is the trend down?
Again, I think people bail on development of these things because they're so expensive and so difficult. You know, they look for, especially the big companies, they're looking for early exits. My medicinal chemistry degree served me quite well on a discovery project working group because a medicinal chemist on my team accused me of killing his compound. And I said, yes, sir, I am killing your compound. That's what my company pays me to do. If it passes muster with what I put it through, then it's probably a pretty good drug. But he was emotional about it. It was his baby. So.
But in 10 years, I mean, it dropped in half. Has that
Right.
become so much more expensive, or what's changed?
Well, I think what this also indicates, and Craig, you might want to comment on this, is that people are going offshore to do their clinical trials because it's easier. And they get into the clinic a lot faster and with a lot less data. So this is all stuff that's happened, you know, in the FDA. We went to a drug device, sorry, not drug device, but medical device symposium we sponsored here a couple years ago. And a guy started the program with to FDA or not to FDA. That is the question. And it was because the regulations have become so, you know, oppressive. So I think that's part of it. I think it's changing. I'm hearing better stories because Congress and the media have really gotten down on the industry and gotten down on FDA. So I'm hoping it will improve, but you make a good point. The trend was down at that point in time. So these are just highlights from 2007. I'll let you read those on your own. But there weren't a lot of new drugs that were coming on the market. And then this is really interesting to me. I take Lipitor, and I'm now on the generic form of Lipitor. But look at the hole in the Pfizer economy. That was in 2012. And that's why Pfizer has shrunk and downsized so many people. It's a hell of a lot of money to make up in your budget. And this is small molecule examples now. That's a statin, right? So generics occupy that space that the big companies hate because they can do abbreviated NDAs. This is all covered under that Hatch-Waxman, it's hard for me to say, sorry, Act of 1984. And all they have to do is demonstrate bioequivalence from a safety standpoint. They do have to demonstrate that they can make the drug similarly, if not better, in the manufacturing part of the process. But why do they cost less? Because they have less R&D expense to recoup, and generic manufacturers don't have to run clinical trials. The vast majority of what I do in the preclinical stages is way less expensive than the clinical phases. I mean, let's face it, it's expensive to run human clinical trials. So here's some examples of things that are coming off the market and I think I use, this is Herceptin, right? And I think I used this as an example. This drug right here is the one that I was alluding to before that's the surprise. This is the Amgen drug. And I think I have put some things in here. Yeah, this is an article from the Burrell report, which if you follow the industry finance aspect of drug development, just before Thanksgiving, I'll read this part because you probably can't see it. Amgen announced that the U.S. Patent and Trade Office issued a new patent for Emeril, extending the patent protection for another 16 years past its 2012 expiration date. This was an arrow to the heart for biosimilars. And there were a number of companies that were lined up to take this on. And if you read down further, Merck was one of them. And they had a couple of companies from South Korea and Turkey, and they were ready to release their generic version of this. And the patent office extended this because they said that by the time they got to selling this drug, they only had 1.5 to 2 years of exclusivity in a 17-year period. And so I think also at this time it was recognized that they were extending patent life from 17 to 20 years. So this was kind of a bridge to that. But that absolutely put a dagger in the heart of these generic companies. So we only have coverage for biosimilars in the generic market for small drugs, whereas the EMEA started doing this in mid-2006. They approved a drug for Sandoz. which if you know the history of some of these mergers, Sandoz was one of the companies that formed Novartis. Sandoz and Ciba-Geigy, both Swiss companies. But we didn't have anything until 2010. So if you blow this up, you'll read that this is passed under the Obama administration as a Patient Protection and Affordable Care Act in March of 2010. And this was designed to give us a similar regulation like the EMEA had already put in place in 2006 to cover biosimilars. Okay, so if you're working with a contract organization, these are some questions that I just have summarized as regulatory guidance, FAQs. Where do I go to find documents? So, you know, you're going to depend either on the contract research organization to tell you where the documents are that they're following, or you can go and investigate them yourself. And you can find most of these online, and I'll show you how here in a second. Which guidances apply to my study? What part does the anticipated duration of clinical treatment play in study selection? Why am I doing these studies, and why am I having to pick a certain time frame for how long I have to do them? And we'll get into the longer term studies post-IND in a minute. And then what role does API or active pharmaceutical ingredient play in terms of its purity in the conduct of my studies? This is a really interesting concept in small molecule, because what happens is that people try to strive for what they're going to put in the clinic. They want a GMP qualified material. And what I always tell them is start with dirtier lots. You know, it's been generically called GLP, and you say dirtier lots and people think, well, what, I got to drag it around the parking lot behind my car? What are you talking about? But you have lesser degrees of purity, and it's okay to test things that are as low, even as 95 is probably the extreme, but you don't have to have 99.99999%. Okay, that's too pure. And when you start with this in small lots of material that you're producing, you run the risk that you're going to scale up and set yourself up for a clinical hold. Because when you start scaling up, things change, and you start seeing bigger amounts of impurities you never saw before. And what puts you on clinical hold is very simple. If you are now giving patients a drug that has an impurity in it that animal testing has never seen, they're going to stop it. They're just going to yank the plug. So I tell people, start with those higher levels of impurities. Typically, we advise like 97 to 99%. And it's okay. You just have to know what the qualification of your impurities are, what percent you are running with. And then what part does the sponsor play in those studies conducted at a CRO? So here's an example of where we can go online at our shop and look for guidances. So most large laboratories like ourselves have the availability to look at ICH guidances, to look at things that you don't see every day, or kind of nuances of, and these are organized, although you can't see it, these are organized under different categories for different types of therapeutic indications. These three right here are all for carcinogenicity. Okay, so this is what the need for carcinogenicity studies is: testing of carcinogenicity for pharmaceuticals, dose selection for pharmaceuticals, and the limit dose. Sometimes when you are testing a biomolecule, you can't test it high enough to demonstrate toxicity. So now you start getting into physical limits. Okay, how much can I get in solution? How much solution can I give in a dose feasibly? And you start talking about maximum feasible dose rather than maximum tolerated. Does that make sense? And so that's both a two-edged sword in the sense that the goal is to demonstrate toxicity, but if you can't, you got to demonstrate how high can I go before I start causing kidney tubule blockage, or I'm precipitating proteins and it's, it's not possible to get rid of it. So every lab should have something like that. So this is the ICH cover page. Anybody in here British? I won't offend you by this, I hope. So it's EMEA, but it's European Medicines Agency. So where the extra letter comes from, I don't know. But this is what the guidance documents look like, because they all have an approval period. Okay, so transmission to, and this is a subgroup that reviews it. Interested parties, so they get to comment. So this is starting in July of 2008, and you go all the way down here to December of 2009 before this gets put out for public consumption, where people are going to actually say, I'm following these guidelines. So here's the M3 guidance, and this is what dictates what you do in animal safety studies, okay? So, and you can see that we started with the M3 guidance back in November of 1996. We now have an M3R2. So there was 2 revisions of this that we're operating under today. And you have a table of contents, so you can go and look at, again, I don't expect you to read this, but you're looking at what the repeated dose toxicity studies should contain or focus on versus acute toxicology. So your acute toxicology are going to be what we call dose range finding studies. Where do I see that MTD? And then how do I ratchet back and test something for lower doses, longer period of time? So by the time you get out to the subchronic studies that cover 6 and 9 months in the clinic, you're testing very much smaller doses than you started with, because you're giving it over longer periods of time. And now you're talking about repeat dose pharmacokinetics and what the steady state, or what the pharmacokinetics people call steady state. Okay, so I take a dose of Lipitor, I've just dropped it from 10 milligrams a day to 5. But that's because I can still maintain a steady state level in my bloodstream that's going to be effective, okay? And so if you're taking something every day for a long period of time, you're going to be at a much, much lower dose, and that's what these guidances are trying to educate you on. Here's the ICH website. This is what it looks like. So you can clearly go on there and find any ICH guidance on the internet. But again, we have access to quick lookups, so we can find it much more quickly in our shop on our own intranet. And then the guidances themselves look like this, and you just pick a category and click on it and go. Here's another one example of it. So the original M3 guidances look like this, and they started, this is back in 1996, and they started with rodents and non-rodents. And if you gave, this is the category here is maximum duration of clinical trial. And if you've given this up to 2 weeks, so even if it's only a single dose, it falls into this category. And, oops, too fast with the clicker. And then between 2 and 6 weeks, 2 weeks and 6 months, same as a clinical trial. So you have to cover the duration of the clinical trial. So you were going a minimum of 2 weeks now to up to 6 months if that's what your clinical trial started with. And then greater than 6 months, you had a 6-month non-rodent, rodent, and a 9-month non-rodent. This used to be 12 months for non-rodents. So that was a pretty expensive, long duration study, but they reduced it in this guidance. And then this is here now to cover, and the categories are covered here. Duration of repeated dose toxicity studies to support phase 3 trials in the EU and marketing in all regions. So you can see even under ICH there are some exceptions, okay? But now what you're looking at is they want to do up to 2 weeks of dosing for this, even if you're out to phase 3, they want you to do a 1 month rodent and 1 month non-rodent. And then it covers all the way up to 6 and 9 months again. So they've revised this. These are the old things, old durations, and this is the new ones into the that R2 revision that I've talked about. So your study director should have a role in all of this. You need to be engaged as a sponsor, but these are questions that a good study director will be happy to answer and live. So what you're doing is you're approving a study plan. You approve and issue amendments to the study plan and document any deviations from them. This is critical. Anything you change in a study design, you have to document in an amendment form. And we have short-term coverages of documents we can sign today to make a change that we can't get an amendment out as quickly. But it's all covered under good laboratory practices. You have to liaise with each principal investigator. So if you do anything even in your laboratory where some other group is doing a measurement, say clinical pathology, you have to have a liaison with those people so that you know what's going on. You are the point of control under GLPs and in the eyes of the FDA. Ensure that all the staff is aware of the requirements and details of the study protocol, and including off-site situations. If you're shipping something to another site, you got to make sure that they understand why and what has to happen and when. You need to visit the study room regularly, ensure final report is prepared, and there is only one study director. We've had some companies come back to us and say, well, we want you to run the study, but we want the study director in our company. And that's like a GLP violation right there. So we say no. And we've lost business because of that, but we have to protect our reputation as a GLP compliant lab. These are some other ICH guidances on toxicokinetics and dose selection. I'll let you look at those at your leisure. But these actually refer to what are called the Misc Guidelines, and this is where you get into drug metabolites. Okay? Now they're starting to be more concerned about, is it your parent that you gave, or is it a metabolite that's really the active substance? Now, in the old concept of small molecules, we talked about prodrugs. So we attached something covalently to a drug molecule and hoped that it got cleaved in the bloodstream because it made it more soluble or it made it more bioavailable. But the active ingredient was actually the parent molecule and not the prodrug. I had one example when I was in Bristol where the parent material was not doing so well, and they said, well, let's roll out the prodrug. And I said, well, it's getting into the brain faster than the parent drug is, so it's not really a prodrug. And it caused single-cell necrosis in 2 weeks in the liver. Now, it was in rats, but that was enough to kill that. So now what they're focusing on, and this was a number of years ago. I've been in my current job now for 13 years, so this is about 15 years ago when that occurred. But this is a guidance that's been issued since called the Misc Guidelines, and it's for human testing of drug metabolites. So there is a whole series of guidances that cover this, but they strongly recommend in vivo metabolic evaluations in humans be performed as early as possible. So the other slide that I showed on phase 0, this is where you would cover that. So we're looking at 5 more minutes, right? All right. So I'll flash through these. But this is where they're focusing on human metabolites raising safety concerns. And it's at a threshold of greater than 10% of the parent drug systemic exposure. So if your metabolite is 10% or higher than that of the parent material in the bloodstream, you've got to address this. And then there's some additional things here that I'll just flash through so we can get to some of the other slides. Now this is phase 0. It typically employs a technique called accelerator mass spec, and that's where you're going to do very low doses of drug, but reasonably high doses of radioactivity to see it. And actually, the doses radioactivity-wise in accelerator mass spec are low enough you can flush them down the sink. It's a pretty straightforward study to run. But you're looking for absolute bioavailability of the metabolite and not the parent. I won't go over this, but this is really the focus of it is on metabolite burden. Clinical ADME studies, this is another category. I'm just going to speed up through this. But you're actually doing ADME in excreta in the clinic in human samples. What would I do there? Oh, good. Didn't think I could do that. Again, this is usually an analytical technique, either AMS or traditional LC-MS MS. And you're looking again for exposure parameters. So this is not really a safety study. It's what's happening with over 10% of my drug being in metabolite form, and is that an active form? Again, here's some more places you can go and look on the internet. This is the FDA guidance for biologics. This is their pages as you flip through them. Here's OECD. I always liked this statement in my protocols, but it says, it is a regulatory GLP requirement to inform the study director about the methods used and the results of these analyses, including any statistical analyses, even if the data will be reported separately. And that particularly involves places where you're sending third-party laboratory samples because the sponsor likes putting their work with that laboratory. But you have to keep in communication, and you have to know what that laboratory is doing. And if they do anything different, and we did have a GOP exception to this, the sponsor didn't like it, but we said, you better tell the FDA now, when you submit this study. They subcontracted the bioanalytical, and we thought they were going to do the pharmacokinetic analysis in that laboratory, and they sent it to yet another laboratory. And they didn't tell us. And so the study director is in violation of point of control. So we wrote that up as a GLP exception, and we got them to calm down and said it's not a game changer. It won't kill your compound. However, had we not pointed that out, the FDA would have been on us for why wasn't the study director informed. Okay, and the document I passed around looks like this. You'll see that I've got a couple pages highlighted where you start with clinical drug design, trial design, and then biopharmaceutics here. And I'm almost to the end. This is our site in Southwest Michigan, It's a 1 million square foot facility near Kalamazoo, Michigan. And we are opening a translational imaging center that we just broke ground for in August of last year, and they just dedicated it. And that's my question slide. So does anybody recognize what that's from? Come on, you guys are Huskies fans, aren't you? This is 2 years ago when they had to play at CenturyLink because the stadium was being renovated, and they beat Stanford. And all the students rushed the field. So that hue in the middle is the gold helmets of the football team. So anyway, questions. I know I went through that last part a little bit fast, but I didn't want to bore you with guidance documents per se, but just where to go to find them. Yes, sir.
How's the FDA dealing with different species than rats and dogs? For instance, we're anticipating our compounded infectious disease will be useful in farm animals.
Sure.
Can we use cattle as a proxy for dogs?
You can. The problem is whether that drug is going to be marketed strictly for humans or whether it's going to be done through the Center for Veterinary Medicine. Those are the typical ways that you test in CVM. But we have relationships with veterinary schools where we can do that, and they can do it under GLPs. So we've done horses, we've done cattle. We do a lot of farm pigs for surgery and medical device applications. Our chief surgeon actually has a success rate of inducing myocardial infarctions in farm pigs, and he recovers those pigs after the infarction 3 out of 4. And other places are 1 or 2 out of 4, so he's got a really good success rate. But again, device is regulated slightly differently. So if you're not going into a veterinary medicine, which is where a lot of human medicines start, frankly, then you just need to be upfront with the FDA about what you intend to do and why you're doing it. So, does that make sense?
Yeah.
Good. So we test everything from mice and rats on the small end of the scale through ferrets, rabbits. Rabbits are not rodents, they're lagomorphs, in case anybody doesn't know that. And they're kind of in between large and small animal, all the way up through dogs and primates and mini pigs. Mini pigs is a great misnomer. A mini pig from Sinclair, which is they put out Hanford, Sinclair, their own design, and Yucatans, and they're 70 or 80 kilograms. That's not mini to me. That's 225 pounds, folks. You get in a cage with them, and they tell you what to do. So. But the Gottingans are really a good choice, and they've become more and more acceptable. And they're very social animals, and if they're suffering from intoxication from your drug, you know it right away. They'll just sit in a corner and not move. And otherwise, they're up and they're communicating with their neighbors. They love to chew on these metal chains. You walk into a Gottingen mini pig room, and you just hear this constant rattling of these chains, you know. And so, you know, it's become more and more acceptable. But that's about the largest non-rodents, and we have a lot of primates. There's, I think at last count, last month, we were up to 1,800 primates on the facility, so. Anyone else? Yes, sir.
Do you have any experience with like a company that has already gone through the IND and early clinical process for like an antibody, but it really hasn't shown what they expected it to show, so they want to create like an antibody drug conjugate or some sort of derivative of that initial already approved compound? How is the approval process for that new compound streamlined at all? Can you build upon the previous IND, or do you have to start from scratch?
Again, take a look at the SBIR and see how old it is, because things change from year to year. But they will look at, I guess they've changed from new chemical entity, they used to call them NCEs, to new molecular entities. And they will look at anything you do to modify that antibody as a new molecular entity. It's just easier for them. They're very conservative. We actually have been approached, and I have some slides here if anybody wants to see them. There are about 6 of them on our new Translational Imaging Center. We've been approached by some of the big biopharmas about helping them find out where their antibody drug conjugates are going, because they're coming apart way sooner than they're getting into their intracellular targets. And so we have ways to label them and follow them in 3 separate entities. But that's the reason that they're concerned about it and will look at it as a new molecular entity, is because they want to know, okay, if you got a payload molecule, or some people are even attaching, multiple payloads. They have If it's a spindle poison, for instance, they have like 12 molecules attached to the proprietary linker. Well, they want to know, okay, are all of those getting into the cell when it gets ingested? You know the concept of this, right? You're going to target cancer cells, some acquisition target on the surface, and then the cell is going to either phagocytize it or pinocytize it, and then the payload's going to get released. Well, what happens if that payload's released elsewhere in the body? Now you got problems that you didn't anticipate, and you have to be able to show them that you know where it's going. So there's your biodistribution factor right there. You know, and what we hope to show with imaging is that you can actually show acquisition of the target and it's not going anywhere else. So, you know, at least from a local environment, it's there, and you know, it may be that there's some toxicity around the cancer cells. But what we're hearing too in that category is that normal cells are taking this up in some cases, you know, because they've got similar targets on the surface. So now you have to start to question, you know, how good is your target acquisition? How specific is it to that cancer versus normal cells? Because let's face it, the reason that, and you got a company right here in town, right? Seattle Genetics got approved for non-Hodgkin's lymphoma. The reason that they found that to be so successful is because they're only killing cancer cells. That's the premise, that's the goal. But I think as we get out into larger and larger post-market studies, they're going to find that other cells are taking this up. When you look at the normal approach to cancer, the conventional approach to cancer, it's cytotoxic agents. And a lot of people on chemotherapy elect to stop because the quality of life is so poor. And they'd rather just go home and spend their last few days than go through this hell, chemical hell. This is a new and exciting field, and I hope they succeed. I hope everybody who's in that field succeeds, because I call it a Trojan horse. It's a phenomenal concept. But again, back to your question specifically, they will call that a new chemical, a new molecular entity. And you'll go through the whole thing. Because that's just the way they do it. And they hold all the cards.
Well, thank you very much for coming, and hopefully maybe you can stay and answer any other questions that people may have. So
Sure.
thank you very much.
Thank you, Elizabeth.